galaxy comes with a number of genomes already built in. That's very convenient because then you don't have to search for them. But not everything is there, far from it. Yeah? So sometimes if you work in an organism that's not human or mouse or the typical ones, you have to fetch the genome yourself. And there are 3 locations where you can go for that. And you can see them here. I prefer Ensembl, especially for RNA-seq data. It is the best choice. Theoretically, the sequence should be the same. They differ in the annotation. Yeah? So that's what they do basically. Ensembl and UCSC, they take a genome sequence and they try to figure out where the genes are located in that sequence. That's annotation. So that will be different between these 3 locations. And since you use the annotations for RNA-seq data, you want to go for the website that has the best annotations. And it's kind of agreed on that Ensembl is the best choice. So Ensembl is this website. It's the European website. UCSC is the American one. Ensembl is the European one. So when you go to the home page, there's a link to downloads. So let's click. And then here in this menu, you can select FTP download, which is what you need to retrieve a genome sequence. And then you can scroll down and then you see all the organisms. There are many pages here, all the organisms that are supported here in Ensembl. So let's go for human here. So you see a lot of FASTA files to choose from. Yeah. And the one that you need that contains the full genome is this one, the DNA. This is only the copy DNAs. These are the coding sequences. These are the non-coding RNAs. These are protein sequences. So the full genome is this thing. Click the FASTA link. And then you typically end up here in a folder that contains a huge number of files. So the best thing to choose here is the primary assembly, this one. That contains basically everything, so not only chromosomes, but also scaffolds, so large sequences, where they don't actually know of which chromosome they belong to. So the primary assembly is the best choice for mapping. So that would be the link you would click. You can see here that there are 3 versions of the primary assembly. You have the SM, the RM, and then the version without any M's. Yeah? The regular version. Which one are you going to use? What's the difference between them? So this is just the sequence as it is. RM and SM means that the repeats are masked. That's what it stands for. RM, repeats masked. SM means soft masked. So in RM, all the repeat regions are replaced by an N. And then in the soft masked sequence, the repeat regions are replaced by small letters. So instead of capital A, T, C, and G, they use small a, T, G, and C. So which of these 3 primary assemblies are you going to use? Well, I always use the regular one. Yeah. Soft masked doesn't matter because the mappers will ignore the small letters anyway. So they will treat them as capitals, so it will be the same. And repeat masked, I don't use it. The only reason that I see why you would use the repeat masked regions is to speed up the mapping, because it will skip over large repeat regions which are always hard to map to. So if you want to speed up the process, then you could go for the RM tool. But know that you will miss things because of this. Yeah, you will not, for instance, be able to study copy number variations because that's exactly where they are in these repeat regions.